The work being done by Professor Kool addresses the question of whether other genetic systems besides the natural one might be functional. His group has designed and synthesized new DNA bases that are approximately one-third larger than the natural ones by the addition of a benzene ring. This xDNA helix structure is similar to that of natural DNA, except that it is wider and more stable. Let's now join Professor Kool as he tells us more about the work being done in his laboratory on xDNA.
How did you come to discover, create xDNA, like expanded, bigger DNA? Because there's different ways you can alter DNA.
Yeah, I mean, for example, you could consider making DNA smaller. And so the problem with that is that DNA is held together mostly by the bases stacking on one another, like a roll of coins. And the smaller the stacking surface area is, the weaker the helix is. So we knew that making things smaller was probably not going to be the right direction to go in, that it was unlikely to be stable as a double helix at room temperature or at body temperature. And so that limited us to things that were at least as big as DNA or larger. And we've looked at some things that are alternatives that are as big as natural DNA. Our group invented non-hydrogen bonding base pairs, which are now used several places in the world in multiple groups by biotechnological purposes. And other groups have looked at rearranging the hydrogen bonds on purine pyrimidine architecture. So definitely there are ways of doing it that are about the same size as natural DNA, so we just said, well, what's the other thing you can do, and that is make things larger. And we didn't see any reason why it couldn't be so. And there was even some literature back from the 70s on making one in which a guy named Nelson Leonard made a DNA base that was larger. He actually made an RNA base that was larger than a natural RNA base. And so he had already kind of developed a general idea of doing this, and so we thought it was something that could be extended to actually making helices, designing variants of all four bases, and the more we thought about it, the more that it might be interesting.
We also read that the more complex DNA system of xDNA can be used to store more high density information. Can you elaborate more on that? What types of information could you encode with 8 bases?
Mm-hmm. One is actual information. So there are people who want to develop computers out of DNA, to do logic out of DNA. Also, people are using DNA to store security codes and things like that. So, for example, right now, people are tagging explosives made in the U.S. with DNA. So it's used as a security code, so you know where it came from. You mix in a little bit of DNA with a known sequence, and then you know it's like a barcode, but it's the tiniest barcode that can be amplified from tiny quantities. But in addition, just logic and things like that can be stored. In a cell, everything that a cell does is stored as information in a sequence of DNA. So there may come a day at which not only can we make a cell from scratch. When I say we, I don't mean me, I mean people generically, people like Craig Venter. But that we may, they may have a new genetic set. They may not necessarily be confined to just A, C, T, and G. And so, for example, xDNA has 8 letters instead of 4 for normal DNA, and you can encode 512 codons. So right now we use, we have 20 amino acids encoded by up to 64 codons because we're limited to 4 letters. So you can imagine a day in which you have 512 or 500 different amino acids to choose from rather than simply the 20 that we use now. Mm-hmm. So it's far in the future, but there is a lot of important things that comes from being able to store information in DNA in many areas of technology, so biological and even non-biological. There's even structure, so people are folding DNA into useful things, doing nanotechnology with DNA. There's a paper that just came out in Nature or Science this week on building a box out of DNA with a closing lid. So being able to encode more complex structures. More complex and larger structures can be done if you have more complexity in the molecules that encode it.
So we understand that xDNA is too large to be replicated by normal DNA polymerases. Is there any work currently being done to make like a larger one to try to replicate it?
Yeah, that's one of our big goals, is to be able to replicate xDNA. So we need to either discover or develop a polymerase that can replicate it. We would love to be able to do PCR of xDNA, for example, to amplify xDNAs, to make long sequences of xDNA. And so, yes, we want to develop polymerases. We're looking around through existing polymerases. Most normal replicative polymerases probably won't be able to handle xDNA. We've tested some, and they're not very good. But some polymerases that are used to handling damaged DNA that's usually mismatched or larger than natural DNA. Damage almost always adds groups to DNA, so it makes it larger. Those enzymes actually are better at replicating xDNA. We haven't published it yet, but we found a couple of enzymes that can replicate xDNA reasonably well. Not good enough for PCR yet. So anyway, we're working on ways of either discovering or developing polymerase that can replicate it, and that's definitely an ultimate goal for this project.
Once you're able to replicate it, what specifically are you looking to answer about normal DNA behavior? What exactly is currently unknown?
Well, part of the reason for doing this project is to ask what about DNA's structure makes it function the way it does. So if we think we understand DNA, we should be able to make our own. So we're testing what we know about what in the molecular structure of DNA makes it a replicable molecule, for example. If we think we understand that, then we should be able to design our own replicable molecule. And so to prove that, we have to replicate it. So part of it is simply to ask the basic science question of what makes DNA a replicable molecule. And to us, the most interesting part of that is the base pairs. So can we design new architectures for base pairs that function like natural DNA does? And to us, the ultimate function is replication. There are lots of interesting things that DNA does. For example, self-assemble into a double helix. We've already done that with xDNA. It works great. The next challenge, the biggest challenge, is can we replicate it? Because that's where storing information and carrying it on works. And so many uses of DNA come from the ability to replicate it and amplify it, like in PCR, that we would love to be able to amplify xDNA and do practical things with it. For example, xDNA is predicted to be a great molecular wire, much better than DNA. It's predicted to carry charge, both positive and negative charge, better than natural DNA does. And so if we could make long wires by PCR, then we could bridge such a wire between two electrodes and things like that. So that's another thing that's in the future of this project.
Like what's the significance of knowing that there are different stable structures of DNA? Like what does this mean about like other possible life forms and like how their DNA or genetic system is designed?
Yeah, there's two interesting questions. One is what is life like on other planets? We're going to discover life one day, I think, in other places. And the question is, what does its genetic system look like? So what should we be looking for in these kinds of molecules? Does it have to have A, C, T, and G? Are there other letters that might be possible, but using a similar kind of architecture? Or could you envision a totally different architecture for a genetic set, like what we're working on with xDNA? So if we can show that xDNA can have all the functions of natural DNA, then that proves, I think, that we should be looking for things other than natural DNA when we're looking for replicable systems that might have evolved sometime in other places. But then there's the question of why we wound up with the structure we have now on our planet. So that's a basic science question too. And so we have to ask, could it only have been A, C, T, and G, or could other things have existed? A, C, T, and G clearly won for some reason on our planet, but the question is whether other things could have been considered.